And the idea is to see in problems in a model developmental system, which is a shrimp shown over here, how you can address what kind of order may be present in the system in a highly non-equilibrium system that can drive later a body template that can lead to the formation of limbs, legs, things, large-scale structure for this organism. And so the talk will focus a lot on the nature of orientational order, first in equilibrium, and then I'll try to convince you that it can actually be promoted by non-equilibrium processes, in particular cell division. This is the organism that we will return to. This is the star of the show, Paheale hawaiiensis, a shrimp that exists in intertidal marine waters all over the place. So in biological systems and others, we want a great deal of flexibility, but we need some kind of structural rigidity. Otherwise, the system would fall apart. So we're left with the situation, if we consider fluids, which are very rich, they tend to be too disordered and too fragile. By the way, this will be all in 2 dimensions, curved, but 2 dimensions. If we consider crystals, as very nicely discussed yesterday by Oog, they tend to be too rigid, don't have the flexibility to lead to the development of templates and later functionalized structure. So you need something in between, some kind of a structured fluid that has rigidity and flexibility. What can this structure be? One candidate that I'm going to discuss in detail is orientational order. Now, we're familiar with orientational order in 3-dimensional pneumatic systems, but there you have a fundamental anisotropic orientational building block. I want to deal instead where you don't put in any orientational bias to begin with. Now, in 3 dimensions, orientational order and translational symmetry, if you want to break symmetries from the fluid state and generate orientational order, it tends to also break the translational order. 3 dimensions, these are highly tied together because if you orient, but you look at it from any possible position, that changes the orientation. In 2 dimensions, however, it's possible to independently break symmetries to produce orientational order without translational order. And this is a phase that's, depending on the kind of the degree of the orientational order, that's called a p-atic phase. The most common P=6 case, discussed yesterday, which is the hexatic. And the shrimp system that I'm going to tell you about actually exhibits a 4-fold kind of order, a hexatic order. What is a p-atic phase? The order parameter is the sum of the p-th roots of unity. And very important, a correlation function. So the relevant correlation function will be 2-point correlation function in space of this order parameter. And we have to simultaneously look at translations. It's a density-density correlation function. So in the equilibrium case, this is the theory of 2D melting, as Oog discussed. And if we go from left to right in terms of symmetry breaking, we start with a fluid. It is disordered with respect to all symmetries, rotations, and translations. And it's dominated by a topological defect, which is a disclination, which I'll discuss in a minute. In the hexatic phase, you break rotational symmetries, you have orientational order, but no translational order. And these disclinations bind into a defect, into a state, which is itself a topological defect known as a dislocation. And I'll show you what that does to the system. And then finally, you can break independently, in the second stage, break translational symmetry to induce translational order. And these dislocations bind with anti-dislocations tightly bound in a crystalline-like state. So this is going, I'd like to discuss it going from fluid to in a more ordered state, because this is what happens in the biological system. Disclinations. Disclinations, for example, we put in a point that has five-fold order in an otherwise coordinated, six-fold coordinated system, then if we parallel transport around such a defect, shown here, you think you're in a regular background, so you want to turn 60 degrees, but you have a mismatch of 12 degrees because the angle of the pentagon is 72 degrees. So you have a 12-degree mismatch, turn again, 24, 36, 48. Finally, you come back rotated by 60 degrees with respect to where you think you started. This means that your orientational vector, bond orientation, is fundamentally frustrated. You don't know whether you're pointing here or here. So if you have a gas of these things roaming around, you will destroy any rotational symmetries. The charge of this defect, five-fold coordinated point, is usually classified by 6 minus the coordination number over 6. So here it will be a 1/6 charge defect. If these disclinations bind to anti-disclinations, so you cancel out the rotational frustration, you get a dislocation, tightly bound 5-7 pair. This is actually a picture from real colloidal crystals from the work of William Irwin. And here's a dislocation. It's given by two freely terminating lines in the crystal. Two Bragg rows, or three Bragg rows become two Bragg rows each side of this dislocation. If you make a circuit outside the dislocation, it will close translationally. If you make a circuit around the dislocation, it won't close. There's a mismatch by this vector, the Berger's vector, shown here, which means, again, you don't know whether you're sitting here or you're sitting here. So you're translationally frustrated, and if you have the gas of these dislocations, then you will lose translational order. System will be translationally disordered. I've been talking about four-fold order, so on a square lattice it looks a little bit different. 3 Bragg rows going to 2 corresponds to something like a 5-3 pair in a square lattice. Now, the correlation functions are the most important thing that will determine which phase you're in here. So for the liquid, you are translationally disordered with respect to translationally disordered short-range order for translations, short-range order for orientations. In the hexatic, and this is what Ug was talking about, you have short-range translational order, so it's translationally disordered, but you have quasi-long range, the only thing sort of possible in 2 dimensions for the orientational correlations. This quasi-long range power law order is a rather subtle thing. And in the crystal, you have quasi-long range translational order, that's a 2D crystal, but you have truly long range translational, truly long range orientational correlation functions. And this actually is what Ug used to sort of classify, to determine that you're in the crystalline phase. This is actually the best way to determine that you have a crystal. So to establish that you have orientational order, you have this twofold task. You have to show the power law behavior, quasi-long range order for orientational correlation functions, but simultaneously that it's translationally disordered. That's crucial and quite tricky. Now for the shrimp, this is what it looks like. It's known as the Swiss Army knife of developmental systems. It's what's called a direct developer. That means that the juvenile system is identical. It just scales up in size with respect to the adult system. There's no larval stage. And so that makes it rather simple from the point of view of thinking about development. It has 17 different appendages, from these feelers here to things here to limbs and legs, all the way along here, that are used for walking, eating, sex, everything possible in this system. How do living systems reliably generate then complex shapes far from thermodynamic equilibrium? This is sort of one of the key questions in developmental biology. I'd like to shed maybe some light on it from analyzing this system. The appendages, of course, have to be correctly arranged with correct replacement and orientation for proper functionality, so you don't want this hand ending up over, this arm ending up over here or ending up as a leg. And if you do some genetic manipulation, you can end up with a limb where an antenna should be. It helps to have a plan to serve as a template for this structure. And there's a relatively simple rectangular looking kind of system, so you might hope to have shed some light on this development of this template for complicated structures in such a system. System develops kind of developmental chronology is very much linked to the spatial organization of the system. So it develops in terms of these kind of layers here, and each layer is genetically switch on development of the associated limbs. Here's more developed, here's less developed as you go down. This happens sequentially. In the Stricon lab, this system was imaged by multi-view light microscopy. And here's a picture of what these things look like. Here's the head A, here's the tail V, and this is the dorsal and ventral lines going around this way. And you can see some kind of fairly regular structures, but the question is, what is the order in this structure? It looks almost like crystal, but you know, if you look in, for example, Cattell solid-state physics books, a lot of the pictures that are supposed to be crystals actually are orientationally ordered phases. Translational order is very, very hard to see, whereas the eye is fairly good at picking up orientational order. So that's what we set out to do. This is the light sheet microscope from the Stricon lab. You illuminate this way and image orthogonally to that. It's a very powerful way of getting, and fast way of getting images. Now, this system is dividing. It has a doubling time of about 10 hours. And so you have a reasonable amount, not as much as in Drosophila, but a reasonable amount of cell division going along. So cell division is a very, very disruptive process, and it's normally expected that cell division will fluidize the tissue. But it turns out that fourfold orientational order is established and maintained in this system. Here's the sort of full view of the system. It's somewhat curved, but enough you can do tissue cartography, project, and triangulate these systems shown here. And then we analyzed the order parameters and the correlation functions. The system has not six-fold order, but turns out to have fourfold order. This is the fourfold order parameter for this system. And this sort of gives you some intuition of what it's like to be orientationally ordered. If you have a system like this, this has six-fold order, but not fourfold order. So the fourfold correlation function, order parameter is quite small. This has fourfold like order, sort of square-looking cells. And this has a high psi4 order parameter. Here's a disordered system, and here's what correlation, a highly sort of correlated, orientationally correlated structure would look like. So we measured this over time, and this is about 3 days after egg laying, measured over about 35 hours, and the psi4 order parameter grows from very small up to a fairly large number, above a half, almost 0.6. And it develops sequentially. You have precursor rows that form. You recruit from the disordered posterior side of the system into a more ordered side, and you then, still disordered, then you develop these precursor rows which grow sequentially and develop into this kind of structure here, which is the structure where it's developing orientational order. Simultaneously, you measure the translational order and find that it's disordered. The correlation function shown here, orientational correlation function, is power law, which fits fairly well to -1/4, which will be the behavior at the exotic to liquid transition point, but it grows sort of continuously through that exotic phase. Yes?
How frequently are the divisions happening? Is it continuously happening?
Yes. Doubling time is 10 hours, so this is over 35 hours. So, yeah. So it's nowhere near as fast as Drosophila, but it's very important for the system. And this is just basically showing the same thing, showing the development of order in the system. Now, how is this possible? In equilibrium systems, these hexatic phases are very, very hard to find. For example, if you look at Leonard-Jones point particles with a repulsive 1/r to the 12th potential, this was studied in the mid-90s by Bagchi, Anderson, and Swope, Hans Christian Andersen, from Stanford, chemist. And the hexatic phase exists for a density range of 1%, between 1 and 1.01. Interestingly enough, the way that they uncovered this phase was to simulate a bicanonical-like ensemble. They simulated n particles, and then they added one particle, simulated, and then removed, and came back and moved between these two ensembles. This gives you very good statistics and gives you ways of exploring the configuration space for the system. If you want to move through the space of all triangulations of a two-dimensional system, there are two basic moves known as Packner moves. One is the bond flip, which preserves the number of vertices, just a flip of a diagonal from this way to this way. But the other one is node insertion. So you put a point in the middle of a triangle and then retriangulate. That's known as the T2 move. This is both of them are ergodic in the space of triangulations, but if you have both, a very efficient way to move through the space of all triangulations. And here, cell division is akin to a node insertion. It's like interstitial in material science, but it's happening all the time. So you're continuously stirring an active system with an extra kind of noise associated with cell division. This creates a complex of dislocations, an interstitial. It's not a defect itself, but it always morphs into a complex of dislocations with net Burger's vector 0, and then it can roam around and find other equilibrium states corresponding to this kind of subtle order. That's the notion that I call here defect-driven morphogenesis. Even in flat space, if you put in an interstitial, this is the actual picture from colloids, put in an interstitial, you see it usually becomes a bound state of 2, a dislocation and anti-dislocation, or 3 dislocations in this kind of hexagonal structure. But these things are interacting, moving around, stirring the system, and when you're continually doing that with division, the idea is that this enables you to find this orientationally ordered phase. So summary from this part is that fourfold order is uncovered in this growing Pahiale embryo, and patterning the timing, locations, and orientations of cell divisions. As they divide, they reorient and establish this quasi-long-range orientational order, serves as a non-equilibrium order-generating mechanism. Now, I'll just mention briefly that we've followed up this picture in work with Max B at Northeastern and a graduate student of his and an undergraduate at UCSB who's now gone on to graduate school. And we study a self-propelled Voronoi model of tissue. So you have activity through self-propulsion, so it's modeling the active forces in the system, but we add, and this is the new ingredient, is to add cell division to this system. And interestingly enough, any amount of cell division actually destroys the crystalline phase in the system. So what happens is, even if you're at very low activity, Cell division takes over as a noise-generating mechanism and destabilizes the crystal. So this is kind of opposite to the situation that you had because of cell division. So what you have is a low temperature. If you move along here, this is cell division rate, and this is the self-propelled velocity. As you move from low activity to high activity, you have a re-entrant phase transition. Liquid, then hexatic in this case, and then liquid again. At sufficiently high division rates or mobility, you're disordered. So this is a fairly large region of this orientationally ordered phase that is enhanced by having these non-equilibrium generating mechanisms, activity, and cell division. In this case, you can get a lot of information about the system, so you can actually see that there's a low temperature crystalline phase, there's a high temperature fluid phase, and in this intermediate hexatic phase, you have exactly what you expect, a gas of dislocations. These are 5, 7 pairs. And in this hexatic phase, you can see the density of disclinations drops down to 0. It's this range here. You have non-zero density of dislocations. So I'll just summarize here that a liquid hexatic and hexatic liquid re-entrant transition emerges from the interplay of cell division and cell motility. You need both. The active forces from cell division can lead to a broad region of parameter space for orientational order, hexatic for the vertex model, but fourfold in the particular case of the shrimp. As a simple mean field theory actually can explain the mechanism of defect-driven morphogenesis. That's in the later work. And these theoretical predictions may help to explain the origin of orientational order during embryonic development. So it turns out that this shrimp system is not only a culinary delight, but it's also a superfluid. Since it's in the same class, this is the same class as superfluid transition, the orientationally ordered phase. So following Lipman Beers and Mark Katz, who asked, can you hear the shape of a drum? You can ask here, could you taste the essence of superfluidity? Thank you. Yeah. Yeah, that's numerical simulation. Yeah.
The new cell, like, because it's randomly oriented, suppose I maintain the nearest neighbor orientation.
Yes. Yes. It's only the position. So you choose randomly.
Position is random.
Yeah.
So suppose I maintain like the nearest cells are in the same orientation. Like one cell when it divides,
Yeah.
it maintain its orientation.
Okay.
The new one and the older one.
Yes.
In that case, what will happen?
I mean, that's sort of forcing the system, enhancing the orientation. Yes, okay. So, yeah, that's a very good question. So in the shrimp system, what happens is that when it divides, it is kind of randomly oriented, and then you find that it reorients later to maintain that quasi-long range orientation order. And that's, yes, that's a highly non-trivial process, exactly.
Yes. The orientational order is probably aligned with the body axis of the developing organism, or is
yes,
somehow aligned to this overall structure. So how does this work? So is this a boundary condition effect, or is there other alignments taking place?
No, and we don't know the answer to that because there's no actual data for that. They only have the positions of cell centers. But most likely, at some point, if I had to guess, there's some kind of actin-myosin cable that's formed transverse to the head-tail axis. And then that sets that kind of template. But we don't know. Don't know at all. Yes, Momita.
Very nice talk. So I had a question about the rate of apoptosis. Is it a fixed rate, or is apoptosis happening based on the state of stress of the cells? Because in actual shrimp, what's happening and what's happening in the model?
Yeah, no, it's fixed, and with each cell division, there's one death to keep the number constant and a fixed division rate. And in the simulations, in the shrimp, yeah, I don't know.
Thank you.
I think it's
Kind of a follow-up of that question. So you just mentioned that one cell dies and it is replaced by another cell. So how does the organism grow then?
yeah, and that is not true. That's true as describing the simulation. In the physical system, that's not true at all. So it goes through various phases where density increases if it's just dividing, and then later it goes through phases where the area grows to keep density constant. So yeah, in the physical system, division is dominating. There's growth. So they have not studied that. That would be a very interesting problem to study in the self-propelled Voronoi model, for example. Actually have growth. It's a lot more difficult.
Yeah, I have a question for you.
Yeah.
You said that insertion of a new cell, of new cells, destroys completely the translational order.
Yes.
But then in your last picture, you still had translational order for finite division rate.
No. Oh, no, yes. Here you mean?
Here, yeah.
No, in this phase here, it's only orientational order, translationally disordered, gassive dislocations. This is the hexatic phase.
Okay.
Is that clear?
Yeah.
Yeah.
And the other question is very naive. How would you define change your model to go from hexatic order to tetratic order? Do you have to change interaction somewhere? Or
Yeah, so
do that?
most likely the tetratic is six-fold order with two very short edges. So if you adjust tensions, so the tensions are high on two edges and shrink them, then you would get something that looks basically four-fold ordered. I think that would be the easiest way. Or you could just start, instead of building on a triangular system, you could start in the simulation, in the Voronoi model, with a four-fold like grid. Yeah, it's not as natural because triangular packing is usually favored. Yeah.
Other questions?
I mean, the four-fold order is highly unusual. For example, in a crystal, you have to have fine tuning with the next to neighbors interaction. That's root 2. So it's one of the things that interested me.
That's exactly what I had in mind.
Yeah.
Okay, thank you very much.